this with a business model. What happens is when you start a business, what initially you need to invest in it, right? So that you can get returns. You invest in the business and you get returns more than how much you have invested, right? Same thing takes place. Right now the glucose molecule is quite stable here. Because of its stability, it is not able to produce any energy. So first we need to convert this glucose molecule into something which is unstable so that we can get ATP out of it or energy out of it. Okay. So the same thing is done. Till here you will observe till this phase, till this 3-PGL. Okay. This is actually the investment phase, and the lower phase what you observe is the payoff phase. Alright, so let's talk about glycolysis. Okay, I'll just write a chart, I will show a chart and then we will discuss about it in detail. Glycolysis, which is also called as EMP pathway. This EMP pathway stands for Emden Mayer of Parnas. These are three scientists who gave it. It is also called as anaerobic phase or pathway of aerobic respiration. Why is that? Because here you will observe that in organisms which are anaerobic, in them also the glycolysis takes place. In aerobic as well as in anaerobic, in both it takes place. Okay, so let's draw it. Alright, so now we will discuss about glycolysis. As I already explained that glycolysis, EMP pathway and anaerobic phase of aerobic respiration are the same. Now we discuss about glycolysis. As I said that glucose converts into pyruvate. Initially we have seen, so glucose converting into pyruvate, right? So this process, if you remember, you must have seen the previous this, then you will see that there were multiple arrows are made between glucose and pyruvate. So why I made multiple arrows? Because of this. There's a complete pathway which actually is responsible for formation of pyruvate from glucose. Now here you can see glucose. Glucose is undergoing various processes, various steps, and results in formation of ATP. You can see ATP, NADH plus S plus. You can also see there is production of ADP here, right? So these all things are actually taking place. So now it takes place in stepwise manner. However, let it be in plants or in animals, glucose is not directly present in form of glucose. In animals, glucose is stored in form of glycogen, and during starvation that glycogenolysis process takes place and glycogen converts into glucose and then it is used. If I talk about plants, in case of plants you will observe that glucose generally is present in form of starch or in form of sucrose, because it is transferred in form of sucrose and it is stored in form of starch. So however glucose is formed. So now what happens is, the thing is, this is just like, you can just relate this with a business model. What happens is when you start a business, what initially you need to invest in it, right? So that you can get returns. You invest in the business and you get returns more than how much you have invested, right? Same thing takes place. Right now the glucose molecule is quite stable here. Because of its stability, it is not able to produce any energy. So first we need to convert this glucose molecule into something which is unstable so that we can get ATP out of it or energy out of it. Okay. So the same thing is done. Till here you will observe till this phase, till this 3-PGL. Okay, this is actually the investment phase, and the lower phase what you observe is the payoff phase. Here we generate energy molecules and here we are investing energy molecules. So what happens is glucose is stable. So now glucose has to be made unstable. So for making it unstable we add phosphate to it. Now phosphate addition will come from where? It comes definitely from ATP. So ATP you see convert into ADP, and you know ATP converts into ADP and IP, but here IP is, that is inorganic phosphate, is directly added or attached, or I can say transferred from ATP to glucose. Okay, and that ATP again means is transferring phosphate. So phosphate transferring enzyme is generally called as kinase, and here you can see that there is glucose, which is 6-carbon sugar, hexose sugar. So it is therefore enzyme used is hexokinase. Similar logic say remaining all enzymes also are named. Okay, I'm not going to discuss in detail about the nomenclature of enzyme. However, you see here there is hexokinase present. Hexokinase enzyme in presence of Mg++ results in attachment of phosphate from the ATP on glucose. Now if you see the structure of glucose, the cyclic structure of glucose, the first member is oxygen and remaining 5 members are carbon, which makes a hexagon. But the 6th carbon is attached on the 5th carbon, which is flanking outside, hanging outside. So because of that, you will observe here that the phosphate attaches or attacks directly the last carbon, which is not in the ring. So you can see glucose 6-phosphate. So one molecule of glucose converts into one molecule of glucose 6-phosphate by addition or transferring a phosphate from ATP. Okay, resulting in formation of ATP. So here you can see here when investment of ATP has been done, you are observing that this cycle actually, or this pathway, is respiration, cellular respiration. So this is actually required for generating energy, but it is utilizing energy. As I said, it was investment phase, their investment is taking place. So now what happens, you see here 6 carbon glucose converts into 6 carbon, glucose 6-phosphate. Now this glucose is isomerized into fructose 6-phosphate. The main reason behind isomerization is because we need to make this molecule more unstable. To make this more unstable, we require one more flanking carbon outside. From where will we get that carbon? That carbon will be actually provided. Sorry, we require, the carbon which we require, flanking carbon, will be provided by converting this into an isomer that of glucose, that is fructose. Fructose, if you see, it's a furanose ring. This was pyranose, this is furanose. So means it has only 5 members in the ring. So 4 carbons, 1 oxygen, 5. And on second carbon there's a carbon, first carbon, and on 5th carbon there's a 6th carbon. So 6th carbon also hanging outside, means this carbon remains hanging here itself, and the first carbon also is hanging outside now. Again, the addition of ATP is taking place to make it more unstable. Generally, if there is more than one phosphate in a compound, that makes it unstable. So here the same thing is happening. See, this gets unstable. So it converts into fructose 1,6-diphosphate. Again, the enzyme uses phosphofructokinase. Well, because fructose already has phosphate. So phosphofructo, and as phosphate is being transferred, kinase. And again here you can see cofactors I have mentioned, Mg++. Okay, so fructose 1,6-diphosphate is formed. So therefore the phosphate is attached on first and 6th position. So you can see one molecule everywhere. There's one molecule yet. Okay. Now here again ATP is used. So 2 ATPs are now utilized. Now fructose 1,6-diphosphate, as it is unstable, undergoes cleavage with the help of enzyme aldolase. If you remember, this is nothing but the reversal of C3 cycle. Okay. So C3 cycle mein the formation of glucose takes place in the same way. Here you can see this is reversed. From here in C3 cycle, from here it was forming glucose, and in glycolysis from here this one. So therefore C3 cycle mein the formation is also called as glycolytic reversal. It means reversal of glycolysis. Okay. However, aldolase results in formation of two separate molecules of glyceraldehyde 3-phosphate, also called as 3-phosphoglyceraldehyde, and it also forms DHAP, means 2 molecules, as in 3-PGL 1 molecule and DHAP 1 molecule. Okay, so both of them are 1 molecule. Okay, now the enzymes present further for glycolysis actually can only convert or only process 3-PGL. Therefore 3-PGL comes ahead for conversion, and this DHAP, that is dihydroxyacetone phosphate, is also an isomer of 3-PGL, that is glyceraldehyde 3-phosphate. This is the aldose, this is ketose. So this in presence of phosphotriose isomerase, as these are triose compounds. Okay, that is triose sugars. So this gets isomerized into 3-PGL. Now you can see here 1 molecule of 3-PGL, 1 molecule of DHAP. Now this DHAP converts into 3-PGL. So 2 molecules, one from here and one from here. So now you can see 2 molecules of 3-PGL. Again, this is stable. You must have observed C3 cycle may when the 3-PGL is formed. That 3-PGL formations, what's the main reason? So that because it is stable and it can divert the pathways. From there only you will observe that the glucose is formed, as well as from there the regeneration of RUBP takes place. So it's stable. So now it has to be made unstable. But this time instability is created with the help of addition of phosphate. But that addition of phosphate is from phosphoric acid, directly inorganic phosphate, not from ATP. So what happened here? See, H3PO4, 2 H3PO4 got added. So what happened? This 2 phosphates got attached to this 3-PGL. Now where it will attach? On the first carbon. So results in formation of a compound 1,3-dipGA. And at the same time, as aldehyde is converted into acid, this is dipGA, means diphosphoglyceric acid. So first and third position has phosphate and it is glyceric acid. Here it was glyceraldehyde. So aldehyde converted into acid. Because of this conversion, what exactly took place? Oxidation, that is removal of hydrogen. So that hydrogen which was removed now will be accepted by hydrogen acceptor, that is NAD. NAD accepts that hydrogen, results in formation of NADH plus H plus. However, this in many books it is written as NADH2 also, but actually the attachment is NADH, single H attached, and which further attaches with H plus. Okay, with a partial bond. Therefore, we write separately plus H plus. However, one molecule of 3-PGL releases 2 hydrogens, which is accepted by 1 NADA. Okay, therefore 2 molecules of 3-PGL will release 4, so it will be accepted by 2 NAD. That is therefore forming 2 NADH plus H plus. See, the first molecule got formed here, NADH plus H plus. However, that resulted in formation of 2 molecules of 1,3-diphosphoglyceric acid, which is of 3 carbon. Now, this undergoes removal of phosphate from the first position, which is highly unstable. Okay, therefore what happens is the phosphate whenever removed, again kinase enzyme will be involved, but phosphate removed will be accepted by ADP. Rather it is removed, I'll say it is transferred. So the phosphate is transferred from 1,3-dipga to ADP, resulting in formation of ATP. See another. Now one phosphate released from one molecule, so 2 phosphate released from 2 molecules, right? So both the phosphates are released will be accepted by 2 ADPs, resulting in formation of 2 ATP in presence of enzyme phosphoglycerokinase, or you can directly write 1,3-dipga kinase. That also will do, no problem. So now 3-PGA is stable, so it has to be made unstable so that further further processing can take place. So 3-PGA is isomerized or mutamerized into 2-PGA, that is the phosphate group which was on 3rd carbon, now is shifted on 2nd carbon. Okay, so it results in formation of 2 phosphoglyceric acid, again 2 molecules. See, 2 molecules trend is going on. Okay, so this actually has to form enolase. It has to form a compound enol. Which enol? Phosphoenol pyruvate. So pyruvate is 3 carbon. Phospho is also present. It is enol. Enol stands for en. En means double bond. Ol means alcohol. So enolase enzyme only helps in formation of enol. Why enol could be formed? Because 2-PGA was present. 3-PGA cannot form PEP directly. 3-PGA can form 2-PGA, 2-PGA can form PEP. Now this PEP further gets converted into pyruvate. So phosphoenol pyruvic acid or phosphoenol pyruvate now releases the phosphate and gets converted into pyruvic acid or pyruvate. Okay, so here again you can see pyruvate is formed, phosphate is being removed or released. So 2 molecules releases 2 phosphates accepted by 2 ADP, resulting in formation of 2 ATP. However, when 2-PGA converted into PEP, there was removal of water molecule. So 2-PGA, 1 molecule of 2-PGA releases 1 molecule of water. So 2 molecules of 2-PGA will release 2 molecules of water. It means there is dehydration taking place. Ultimately, pyruvate is formed. Now this pyruvate will enter in mitochondria. So this is actually the complete process. Now if you see, in first step there was attachment or addition of phosphate, also called as phosphorylation. See, phosphorylation, it is taking for the first time in this pathway, therefore called as phosphorylation 1. Next step, if you observe, there is isomer formation. See, glucose converts into fructose, therefore double arrow, reverse arrow, because they are isomers. That is called as isomerization. That step is called as isomerization, and that is first isomerization taking place in this pathway, therefore isomerization 1. Then fructose 6-phosphate converts into fructose 1,6-diphosphate, where phosphate is being added. So this means here there is phosphorylation taking place, the first one. Here again there is phosphorylation taking place, that is second one, phosphorylation 2. The fourth step you can observe, because of instability, fructose 1,6-diphosphate splits into 2 different molecules, 1 molecule of 3-PGL and 1 molecule of DHAP. So this is what they are getting cleaved. The fructose 1,6-phosphate is getting cleaved, therefore it is called as cleavage. The fourth step. In fifth step, you will observe the isomerization is taking place here. DHAP is isomerized into 3-PGL. So again isomerization 2, right? These steps might not completely synchronize, but understand the meaning. This here, okay. After this, you can observe that phosphate is being added, so phosphorylation is taking place. Along with that, what you can see, hydrogen is being removed, so oxidation is taking place. Dehydrogenase enzyme is present. See. So oxidation and phosphorylation, the next step, 6th step. Okay, after this, see, payoff started. So you can see first time here ATP is generated, so ATP synthesis 1. Then isomerization takes place. Mutamer means it's a kind of isomer only. So 2-PGA is mutamer of 3-PGA. So therefore again isomerization, already 2 have been occurred, so 3. then 2 PGA converts into PEP by removal of water molecule, that is nothing but dehydration. See, dehydration. This is also reversible step. This is also reversible step. And finally, again ATP is being produced here, so you can see there is ATP synthesis too. So this is how. So net gain, if I calculate for glycolysis, you will observe that net gain rather I would say 2 ATPs invested. Okay, 4 ATPs produced. See, 1, 2, 2 ATPs invested here 2 ATP and here 2 ATP produced. Sorry. So 4 ATPs produced. So net gain of ATP is how much? 2 ATPs produced. Right? Also you will observe 2 NADH plus H plus also produced. So this is the net gain. Okay. So general reaction also we write, that we will be discussing in next lecture. I hope I have justified the topic completely. Thank you so much.